To introduce our first speaker at the symposium this morning, I'm here to introduce you to Clyde Hutchinson. Clyde was integral to many of the key advances in the field of genomics over the past decades. Genomics is the study of our chromosomes and the messages that encode life. And so it really doesn't get any more important than that. So to have sort of an icon of this field here to speak to us today is really a fantastic opportunity. And with that, I'll turn over the microphone.
Thank you, and thanks to Larry for inviting me to this amazing event. I'm happy to have the chance to tell you about work that's been going on for more than 15 years at the Venner Institute in our synthetic biology group. And it's work on learning how to design bacterial genomes. So we started this not because we wanted to design bacterial genomes, but because we're interested in the very old question, what is life? And I mean that roughly in a modernized version of the way Erwin Schrödinger meant the question. That is, how can we explain life in terms of laws of physics? And by life, I don't mean anything esoteric or silicon-based. I mean central dogma life. DNA goes to RNA, goes to protein as it exists on the modern Earth. Now, very recently, almost 25 years ago, we began to get sequences of complete cellular genomes, bacterial genomes at first, and as you know, now human genomes and so forth. So if we look at the question, what is life, through the lens of genomics, we can reframe the question as what genes are necessary for life and what are their functions. So our approach is kind of old school. We're less interested in big data to understand complicated systems, but we're interested in simple models. So physicists had the hydrogen atom, early molecular biologists, that's my thesis advisor, Bob Sinsheimer, had very simple bacterial viruses. And now we have simple bacteria. And Harold Morowitz made the proposal that a good model to study simple cells would be the mycoplasmas. The smallest of these have the smallest genomes that we know for any cell that can be grown in pure culture without other living cells present. And they have about 500 genes. Now, it's important, I think Harold Morowitz thought mycoplasmas were primitive, but it's important to realize that we now know that hydrogen atom and the mycoplasmas are simple models for opposite reasons. So hydrogen atoms build up in supernovae to higher elements, up to as high as uranium. But mycoplasmas have evolved towards a minimal cell. They've started out from gram-positive bacteria that have typical sort of 2,000 gene genomes. And they have evolved by loss of genes that they don't need in their very rich environment of a mammalian host, for example. And so they've lost genes to become close to minimal. Now, of course, not just synthetic biology, but we tend to think of the cell as containing a program that's encoded in the sequence of the genome. And then that is sort of the operating system if we make a crude analogy to computers. And the cytoplasm and other parts of the cell make up the hardware that executes that operating system. And the genome and the cytoplasm are lifeless without each other. And of course, the main difference between a cell and other computers is it makes copies of itself. So we, in trying to pursue the idea of what's the genes are really essential for life, we started to look at what was the smallest genome that we could get. So we this is a Venn diagram. It's not two DNA molecules. Okay, so if we consider any bacterium that's naturally occurring, it has a set of core functions that make the cell alive and a set of adaptive genes for adaptive functions that allow the cell to grow in a variety of conditions. And so there are two approaches that one can take to getting rid of those adaptive functions. And the reason we want to do this, we want to have something simple enough we can try to understand it. That's more important than really getting some theoretical minimum. So on the right is a traditional approach where we start with a genome and make a series of deletions in it sequentially to produce a reduced genome. For example, the E. coli genomes produced in the Blattner group. On the left is shown the approach we took, which one can argue whether it's top-down or bottom-up, but the construction is bottom-up. We took a genome and then based on what parts we figured were necessary, we designed the pieces to make a small genome, which could be synthesized completely by chemical synthesis and then assembled and installed into a cell to produce a cell controlled by that genome. And we call that a synthetic cell. And as I'll point out, we don't do this just to irritate people, but I'll describe why. Someday it will be possible, I'm sure, to put a cell together from parts, entirely from non-living parts. But now we don't know how to do it. And this may be the time to try this seriously again, but we don't know how to do it now. So we took an easy way out. We built a genome and installed it into a living cell to produce something which, as I said, we call a synthetic cell. Why do we call it a synthetic cell? This is, I guess it's a Gedanken experiment. So on the right is what we do. We put a synthetic genome, the blue genome, into the red cell with the recipient genome, and we let it undergo divisions. And its cell grows indefinitely. So if you look at it a while later, say 30 doublings after you've lost the recipient genome, and we select for the donor genome, the synthetic genome, by growth in the presence of a drug that genome is resistant to. So after 30 doublings, you have 1 billion cells. And since the original cell only had 1 million protein molecules, very, less than 1 in 1,000 of the cells will have even one molecule of protein derived from the original cell. And the same thing happens if you were to assemble from parts. If you could build a cell from parts with a synthetic genome, and the parts could be the things coded by that genome, or they could be from some other cell, so long as when you put it back together it worked. If you grow that out, you get the same thing. And so any properties of the cell that are genetically determined are the same for these two. And we think that for these simple bacteria that all the properties are genetically determined, although that's, of course, open to argument. So our target for the minimization is a Mycoplasma that's bigger than the smallest one known because it grows faster. It's much easier to work with in the laboratory. So it starts out with about 900 genes, and it's a mycoplasma, so very simple, no cell wall, single bilayer membrane. And the downside of the system we've been as the way we've been using it is we grow it in an extremely rich medium that's not chemically defined. But we're working on that. We're making good progress towards getting our everything working in a minimal medium. And when we started this out, we had some funding from DARPA, and they're engineers, and we think of ourselves not engineers. And they, well, to humor them, we made a design-build-test cycle. And it turns out, of course, this is a very good way to think about it. And so we start out by designing a genome. And to do that, we have collective knowledge of molecular biology, but we found that that wasn't enough. We needed objective tests for which genes are essential. And we used disruption with transposon, which is a piece of DNA that can insert randomly into the genome to identify genes that resulted in death when the transposon's inserted. And then we synthesize this genome by making a set of reusable cassettes and subassemblies, and we can mix and match these. They overlap in sequence at the end, so they can be put together by genetic recombination. And then we found that we could clone these whole bacterial genomes as an extra chromosome in yeast, which is really critical to the way we've done things from this point on. Okay, and then we can isolate the genome from yeast, put it into a cell that's a different cell, has a different gene, but related genome, and select for genetic markers on the synthetic genome. And when we do this, we wind up with cells that have only this synthetic genome. So I just go through this in a little more detail. The design stage. The main tool, as I said, that we use is the machinery from the bacterial transposon TN5. We don't actually use the transposon. We use a puromycin resistance gene from E. coli, I guess. And at the ends, we put the machinery, the sequence that's recognized by the TN5 transposase. And if you mix transposase is a commercially available product. So you just take this little piece of DNA, mix it with transposase, and you get something called the transpososome. And this embodiment of this thing has been done by Li Ji Sun under Ham Smith's direction. So we introduce that into the bacteria, and we collect colonies from cells that have the synthetic genome, and which is selectable by a drug resistance marker. And just for those who aren't biologists, we actually keep the lids on the plates. So we then pick these colonies. We pick a lot of colonies, like maybe 100,000 colonies, and grow it up in liquid culture. Well, we take a sample, save a sample not grown up, and then we pass it several times in liquid culture so that it's gone maybe 50 doublings altogether. And the reason for doing this is we want to select against things where the transposon insertion has allowed the cell to live, but it grows worse, slower. And then we take this whole population of cells and we simply cut up the DNA, and by a procedure that is probably familiar to many of you and is irrelevant to many of you, we are able to sequence the junction between the genome and the transposon. And since we know the sequence of the genome, we can tell where each of these is inserted. So every little triangle, black or red, is an insertion event. And this shows a part of the genome with 3 genes, shown in yellow, blue, and whatever, gray, I guess. And every little triangle shows the position of an insertion. And the P0 inserts are the original population of bacterial cells, and the P4 inserts are from the ones that's been passaged 4 times. So you see there are basically 3 classes of things. There are essential genes in which you do not find colonies growing up with insertions in them. Not because there weren't insertions, but because the insertion caused death. Now, in the other extreme, there are non-essential genes where you see lots of insertions, both in the original population and after many generations. And then there are genes we've chosen to call quasi-essential, which you recover inserts in the original population, but not after extended growth. And these are 2 kinds of things. There are things that grow slowly, and there are things where there's enough of a gene product made so that you can undergo deletions. You still recover sequences from the junctions. So summarize this, about half the genes are non-essential. The rest are either essential or quasi-essential or borderline. And we decided in our minimal genome to just get rid of the non-essentials because we wanted to wind up with something that grows well, that's simple enough to understand. And of course, this set of genes is essential, but the essential genes are necessary but not sufficient for life. So it's kind of like airplane engines. You can lose one and the plane flies, but you lose all of them and the plane crashes. Now, we expect fewer of this kind of problem. The existence of these redundant genes for essential functions was one main problem in getting to a design for a minimal genome. And we expected many fewer of these in mycoplasmas compared to other bacteria because it's been evolving towards a minimum set, but has retained genes necessary for life. And we applied a very simple set of rules to design the minimal genome. We took contiguous clusters of non-essential genes were deleted, along with any intergenic regions between them. But we left the regions, the intergenic regions, flanking the cluster. And any parts of genes that overlap with an E or I gene are retained. And we found it useful to make two assumptions here. One is that when two genes are transcribed divergently, the intergenic region separating them contains promoters in both directions. And also, if a deletion results in converging transcripts, we put a transcription terminator in there if it wasn't already there to avoid transcriptional train wrecks. So we designed a genome, and it was about half the size of the original genome. And I think I need to maybe pick up the pace a little bit here. We built that genome, and we built it, and Dan Gibson, who was at JCVI and is now mainly at Synthetic Genomics Incorporated, one of Craig's for-profit companies, he was responsible for the assembly. And zoom in a little bit. It's interesting. It's put together from little pieces made on a conventional oligosynthesizer that you can order from IDT and build these things. And they're assembled into 1.4 kb fragments enzymatically. And then those pieces overlap, and they can be assembled in vitro by the procedure called Gibson assembly to make pieces of 7 kb. These are color-coded, these boxes, in a way that matches with the rings on the outside here. And several of these 7 kb pieces are put together to make 1/8 genome molecules, and these are scaled up, amplified by rolling circle amplification, and then the whole genome is assembled in yeast. And former student of mine in North Carolina who came to JCVI, Gwen Benders, figured out how to propagate the bacterial genomes in yeast. Essentially, you have to put in certain yeast genes. A centromere, yeast origin of replication is useful, and some marker that you can select for growth in yeast. And there are many ways you can do the assembly, but the most extreme is you can take as many as 25 overlapping pieces, one of which contains the yeast factor, throw them in, and homologous recombination in yeast will produce intact genomes at a rate sufficient to work with. And Vladimir Noskov and Ray Chuang worked out a way that we can produce each of the design pieces in pure form and each of the wild type pieces in pure form. And then we can mix and match those to build combinations in case some of our design pieces work and some do not. And then we have to test the thing. So Carol Lartigue with Nina Perovich and John Glass worked out quite a while ago, 2007, a method for we call genome transplantation, which so far only works in the mycoplasmas we've tried. We would be interested in someone giving us funds to really explore trying to do this in another system, but haven't been able to so far. So the genomes are released from yeast by lysis in an agarose plug and introduced by PEG-mediated DNA transformation into the cell. The cell is a different species of mycoplasma than the one the genes are derived from, and it's different enough. It's easy from a single Sanger sequencing read to tell which genome it's from. And we use a tetracycline resistance marker on the synthetic genome to select for it. So after 30 cell divisions, each cell, it's the same argument I made before. The cells all have genome. All the proteins are made by the synthetic genome. So we pick single colonies, and we can sequence or whatever. So make this to get over a bunch of stuff that's interesting at some level, but laborious to discuss. We went through 3 cycles of this design, build, test cycle. Well, first we made something just based on what we guessed would work, and it was a dismal failure. Only one of the 8 pieces worked, and it didn't give a cell that grew very well when mixed with 7 pieces from the wild type. And then we went through 3 rounds where after each round we would take the design and we would do transposon mutagenesis again. So, for example, our first design, each of the 8 segments grew well when combined with 7/8 of the original genome. And we could make certain combinations of 4 or 5 of these. We take one and do the transposon. We go to the next round. And at the second round, we got combinations where 7 of the 8 pieces would grow together. And the third round, we finally got something where the design, when put together, worked. Okay. So here it is. Here's the result. Yo Suzuki made this display of the result. So the genome is about half the size of the starting genome. It's smaller than M. genitalium, which is the smallest known genome for a naturally occurring cell. And it grows 5 times as fast as M. genitalium, or a little better than 5 times as fast. So what have we got? Chuck Merriman has been involved mainly in analyzing what we have here. So one thing is that all the identifications of the genes are done bioinformatically. That is, by comparison of the genes with genes from other organisms, many of which have had biochemical characterization of the genes. So there's some genes which we call equivalogs, which we have a very high certainty that bioinformatic identification is correct. And then there are 2 categories that we still are pretty sure what things are, but of progressively less certainty. And then here's the surprising, maybe depressing, I'm not sure, surprising thing to us is there are a lot of genes that we couldn't make a definitive identification of. There are about 84 in a class we call generics, which are mostly things like methylases or transporters, where we know what class of function it is, but we don't know the precise substrate. Then there were 65 that we don't know what they do. And in many cases, no homologs are known. So all those are real genes, however. Here we have all the genes laid out in an order that's related to this certainty, confidence in the annotation. And they all match genes from other species of Mycoplasma outside mycoides. And very large number of them match other bacteria. Some, as you see, match things up to human or even to archaebacteria. And those are the ones we're unsure of the confidence of. You can also look at this in terms of what the functions are that correspond to the annotations. So the biggest group, of course, are involved in expression of gene expression. So transcription and translation of gene products. There's a lot happening in the membrane. It's small, so it's a high surface to volume ratio. It imports a lot of things from the medium because it's dispensed with a lot of its cytosolic metabolism. And here's the bad part again. There's a significant fraction of the genes that are unassigned. How am I doing on time?
A couple more minutes.
Till what point? Till 45?
We need to be starting the next speaker.
Oh, okay. Okay. So, the other thing is it grows funny, and there's been a loss of regulation of cell morphology. And it's missing two genes that are known to be important in cell division, but putting those genes back by themselves doesn't fix this problem. And taking those genes away by themselves doesn't cause the problem. So there's a lot that we can think about after this. The minimal cell concept is a simple concept, but to actually put it in practice, we had to look at a more complex situation. So we had to include the quasi-essential genes to get something that grows in a useful way. And we've got a good approximation, I think, to a minimal cell, which would be a good tool to investigate the properties of minimal life. And, you know, maybe one day this will be useful for something. I like to believe that understanding things is useful. So our vision is to have a cell where every gene's needed for robust growth, and we know the biochemical function and biological role of every gene product. And it should then be possible to build a predictive model of the cell, and such a model would, in some sense, show that we know how the cell works. I'm going to skip over a number of things. We're collaborating with the Zan Luthi-Schulten's group at the University of Illinois to do modeling. We have a paper under review for the metabolic model, and that the group also does coarse-grained spatial models and all atom and does molecular dynamic models. And the long-term goal is to try to have an all-atom model of the cell. Of course, that you realize is a way off. And would that mean we understand the cell? Well, it depends on what the model did. I mean, the model should fold all the proteins and everything, but the model we're working on now may not be the final objective, but it would be a great next step. And these are just many of the people who've been involved over the years in this project. And we'd like to thank those who funded the work also. Thank you.
So Clyde, you didn't talk much about what kind of medium this minimal cell requires. It probably, you have to put in all the amino acids, but...
You can assume everything's there. It's a complex medium. It has yeast extract, it has serum that have lipids bound to them, which is probably the reason the serum is important. But there are, in the old literature, there are some defined media for some mycoplasmas. And we've gotten one working now with the starting cell for this experiment, and we're now figuring how to get it a really defined medium for the minimal cell.
Another question right behind you here.
In your last slide, you talked about the possibility of a model of the cell down the road. And I'm just wondering how many years away you think we are from a working model of the cell, not at the atomic level, but just at
I mean, it depends we can make models. You can make models that look like the cell when you make a little movie of it. Now, the Luthi-Schulten group has made models of ribosome biogenesis in E. coli. And I think we're not many years from having coarse-grained models of the whole minimal cell.
So you must have taken that strain that grew 5 times slower and put it in the undefined rich media and begged it mutagenically to grow faster. Did you do that?
Yes, we have some collaborators at UCSD who are trying to do that, and it's not clear. It's just going on now, so we hope so. But
It's funny that you ended up with the same number of genes. 500 genes.
Well, it's a bit less. So the genitalium's about 525, and this is about 470, so it's smaller. So it shows, I think, that genitalium is pretty close to minimal. But the thing is, we can work with this much better because it grows much faster.
I think we have time for one more talk.
Hi, I'm very interested in the approach that you took for defining the minimal set and still found 17% of unknown function. And it's clear when we look at comparative functional genomics that about 20% of our most well-studied genomes have 20% of totally unknown function, can't even be generically described as transporters or anything else.
Right.
And in those cases, I know working with the human sets, that we can define actually new domains and group them into families. We still don't know what those domains do.
Yeah.
And so this is very interesting. And I wondered if you had done some domain analysis in these.
Thank you for bringing that up, because I usually try to make that point very explicitly. And there is no cell where we know what all the genes are, what they all do. And there are a lot of unknown genes in this cell, but the absolute number of unknown genes here is very small compared. Say E. coli, maybe 1,700 genes that don't have good solid annotations. And here there's some of them are, some are essential and many are quasi-essential. That is, you could have some growth without them, but we're keeping them in for the time being so that we have something we can work with experimentally.
Well, let's thank Clyde for his talk. Thank you, Clyde.
Thank you. Thank you.